Martin Schiller, the principal investigator of the Schiller Laboratory of Applied Bioinformatics. We are located at the Nevada Institute of Personalized Medicine at the University of Nevada, Las Vegas.
Hi, I'm Ronald Benjamin, a postdoctoral researcher working with Dr. Schiller. I helped him on this gene editing project. Professors Mira Han from University of Nevada, Las Vegas, and Professor Bing Yang from the Donald Danforth Plant Science Center helped prepare the paper. Sarah Trimidal is an undergraduate who built the database. She was assisted by undergraduates Jiyun Wei, Elizabeth Kong, Adam Singer, and Tyler Williams. This review evolved from two papers published by our laboratory, a 2015 PLOS One paper entitled Damaging the Integrated HIV Proviral DNA with Talons, and a 2016 Human Genetics paper on gene editing.
The team built the database of gene editing scars from the literature. They annotated over 6,000 entries. During the process, we noticed that some papers had very large differences in the number of insertions relative to the number of deletions. We also noticed that the lengths of these edits varied widely, and this depended on the type of gene editing technology used or on whether certain genes were overexpressed or deleted. We realized if these patterns could be identified, they could be leveraged to obtain desired results in gene editing experiments. So this led to our central question of this review. Can designer indels be tailored by gene editing? DNA in the cell can be broken by several environmental sources. Ultraviolet light, radiation, chemicals, or even during replication. It produced both strands getting broken in the DNA. It's very important that this gets repaired with high fidelity because so many breaks happen every day in every cell, and it's important to maintain the efficacy of the hereditary information being retained. Now, scientists have taken advantage of this in several ways, and one of the most exciting ones is gene editing. So into these cells, we can introduce one of 4 types of genetically engineered endonucleases, and they'll cut the DNA at a specific site, and then the endogenous repair mechanisms will take over. One type is meganucleases, another is CRISPR-Cas, zinc finger nucleases, and talons. So once the breaks are introduced into the DNA, how do they get repaired? There's a pretty high fidelity repair system called homology dependent repair, where it uses one of the 2 DNA strands to help correct the other strand. This rarely makes errors. If this fails, the cell invokes non-homologous end joining repair. Now, this can correctly repair the DNA, but often it will chew back the ends or do insertions, and then the repair contains errors in it. This is used by gene editing. There are 2 main repair pathways at work for non-homologous end joining: the canonical pathway and the alternative pathway. Okay, so let's take a closer look at the repair mechanisms. So in non-homologous end joining repair, we could have a deletion where an exonuclease comes in and chews back the DNA, and then it's joined by a ligase. We can have insertions where a new template-independent double-stranded DNA is synthesized before ligation. Or we can have complex indels where we have both events going on, where we have some DNA being chewed back and an insertion, and this can happen more than once. So collectively, these insertions, deletions, and complex indels are all different types of indels. So in the deletion, both repairs pathways have different types of exonucleases which chew back the ends and then ligate it. For insertions, different types of DNA polymerases or terminal transferases insert bases and then the ends are ligated together. So these are producing indels. Both repair pathways, alternative NHEJ and classical NHEJ, have both insertions and deletions. And the lengths vary. Although here we're showing about 10 base pairs being deleted and inserted, you can have as little as one base pair being inserted or deleted, or hundreds of base pairs. And you can even add in an exogenous DNA that can get ligated into the site. So the types of indels and their lengths are fairly random, or thought to be random in the cell. So this presents kind of an opportunity. We're interested in gene editing as scientists, and there's lots of reasons for doing that. But if we can figure out how to select what type of indel, whether it's an insertion, deletion, or complex indel, and then the lengths, and then increase efficiencies, this could help scientists better use gene editing technologies. And it's the subject of our review paper. So one way it could be used is to make new model organisms. One can envision that if you didn't want to create a frame shift mutation, you would want indels of size 3 so that the coding region doesn't go out of frame. So it would make the process more efficient. If we also better understand signatures and how indels lengths and types are controlled, it would help us with interpreting the editing history of genomes during adaptation and speciation. And finally, we could target specific gene elements with gene editing. Okay, so what our lab did is we built a database of about 7,000 edited scars from the literature, and then we just analyzed what happens when certain genes were deleted or overexpressed, or if an endonuclease was introduced. How did this affect the repair process? And here we're just looking at deletions versus insertions. And you can see that some deletion of some NHEJ editing genes vastly increases the number of deletions, while others favor insertions. So one can envision where if you transiently expressed a DNA that affected expression of these genes, you could control the type of indels. Some examples shown here is if you deleted pol lambda, one of the template independent polymerases, then you would get more deletions than insertions. And vice versa, if you overexpress the same enzyme, you would get more insertions. Now, we can also control the length, and we analyze the data and here show all of the different types of gene manipulations and what lengths they favor. So if you wanted really short insertions, you could delete any of the 3 ligases in the 2 pathways, and this would favor shorter insertions. So you could do this to kind of select for the size of insertion or deletion that you want. Likewise, using different genetically engineered nucleases or different architectures can affect whether you get insertions and deletions and their size. So if we look here, suppose you wanted 8 to 25 base pair deletions, the zinc finger nucleases would be the best approach. So in summary, we think that there's a new opportunity in science here to better learn signatures produced in indels for both gene editing technology and for interpreting changes in genomes during evolution and speciation. There's 4 different types of gene editing nucleases that can be used. We have double strand breaks that are repaired by 2 mechanisms, and then we can choose either knockdown, knockout, or overexpression of different sets of genes to select for small, mid-size, or large deletions or insertions as well.